So, Jeff, would you like to start with your presentation and then we can move on to the panel discussion?
Sure.
Yeah, I'll do that.
Yes. I was trying to get out of using the podium, but but unsuccessfully. Thank you for the opportunity to present this pilot project to you. This was actually a project that we've been working on with several different organizations over the past few months to try and really take stock as where we are in the field, essentially. So this is intended to be a pilot project. It's focused on the ability to extract different endpoints from different data sources by asking a similar question. And I think we had a real unique opportunity to have several diverse participants in this. So when we designed the study, we didn't have the benefit of the decision tool that's the topic of discussion today. So I guess I'll leave it up to the audience to be the judge in terms of whether we should have done things differently based on this decision tool or not, and would be happy to discuss how it might fit into which particular pathway here based on the observations that we were able to see. But essentially, what we tried to do as part of this pilot project was to bring together diverse different data sources and better understand how they would ask a similar question. And that question was, what outcomes can be evaluated for advanced lung cancer patients for treatment with immune checkpoint inhibitors? And these are arguably one of the most exciting new products in oncology. The reason that we chose this as a focus point was for that reason. These are being widely used both in lung cancer and a variety of other oncology settings. They have a transformative potential in terms of benefits that have been observed in their clinical studies to date. There's large populations, so we thought that would aid in the feasibility of this type of pilot project. As well as numerous different drugs that are approved for this indication. So our intention was not to re-adjudicate the clinical trial, so to speak, or look at the comparisons between the different drugs. It was really meant to be about the different data sources and what could actually be extracted and what the strength of that evidence would be. So the three objectives are listed here. One, to look at the demographic characteristics that were able to be obtained from the different data sources that participated in this. The ability to look at different endpoints, both in terms of traditional clinical endpoints like survival or progression-free survival, which are typically used in oncology, but also explore the potential for other endpoints, things like time to treatment discontinuation or time to next treatment, to understand how they might relate to more traditional clinical measures. And third, to try and look at whether those were reasonable indicators of overall survival. And you'll see listed here all of the partners that generously lent their expertise and participated in designing this study and analyzing their data. So we conducted this in a distributed manner, where each of the partners here maintained their data sources. And we worked very hard to arrive at some common data elements and methodological approaches so that each organization was extracting the information and performing the assessments in a similar manner as possible. So I really want to thank the representatives from CODA, from Flatiron Health, IQVIA, Kaiser Permanente, the Cancer Research Network, a collaboration between the Mayo Clinic and Optum Labs, and PCORNet run through the University of Iowa site as the 6 participants in this particular project. And I won't go through all of these since these will be made available and these slides are public already. But I think it's worth stressing one thing that we found out very early on, where while some of these endpoints might seem a little bit obvious in terms of how one would define them, or they're commonly referred to in these conversations, but I think it speaks to the future of trying to look at ways to standardize different methodology and how to not only define the endpoints, but how to extract them from different data sources to be able to do so in a consistent manner so that we're comparing apples to apples. But you'll see here the different endpoints that we asked each of the participants to extract from their data sources, both in terms of those that were pulling information from claims data, which encompassed the top 3 endpoints: overall survival, time to next treatment, and time to treatment discontinuation, and for those that were extracting data from electronic health records, where you would need active input of the information in order to assess real-world progression or time to progression from an electronic health record. And the purpose of this wasn't necessarily to deploy all the capabilities that each of the different participating organizations are able to do with their data set, but we tried to identify sort of a common denominator and look at things that each of the participants could pull from their data sets in order to be able to run these analyses that I mentioned earlier, but to try and strengthen them by looking at running them 6 times over to see how well they compared to one another. So in this first table, what I think we were able to demonstrate in a very rapid manner. So, I do want to also stress that the groups that participated in this, did so in a very active and rapid manner. So that what we wanted to do with this pilot project was not necessarily design the perfect study, so to speak, but to look at how quickly we could extract some of this information. So the timelines that we were essentially looking at here, in terms of developing this overall project, we met regularly with each of the 6 participants, probably over a 6-month period, but about a 2 or so month period for them to actually run these analyses, which I'll describe in a little bit more detail. But this first slide demonstrates what we thought was really great consistency between the different characteristics. So we've de-identified the different data sets here, indicated just by letter in each of the graphs here. But as you just sort of quickly look over between the different colors, you see a relative consistency in terms of the ability to be able to identify the population who is actually receiving these drugs quite well, whether it was whether the information was distributed by age, by histology in terms of the type of lung cancer that they were diagnosed with, by sex, whether it was males or females, or by line of treatment in which the PD-1 inhibitors were given to these patients. And the areas throughout the presentation here, the area of missing data, really just represents where additional curation was necessary by that participant. And some of that work is underway. It just wasn't able to be completely done in the times of trying to get this information out. So, again, I think we were it was very interesting that just from a level setting activity that the populations were extremely similar, no matter the source or the size of the data sets that were being examined here. In this next table, we looked at these are the different endpoints, or at least a sampling of that I mentioned earlier. And I think this begins to even demonstrate some of the challenges associated with things that might seem obvious. Even extracting overall survival data is a challenge just due to the availability of accurate and timely death records. So as much as we're talking about complex analyses for real-world data analysis, something as simple as is the patient alive or did they die is a limitation for conducting some of these studies. But I think with that in mind, and that's what's represented here, using 2 different methods in terms of whether the data was readily available from Social Security data or the National Death Index versus other methods of trying to extrapolate a date of discontinuation due to death. But I think overall, while there are outliers for some of the data sets, we at least saw relative consistency in terms of the ability to demonstrate the magnitude of overall survival that was seen by the assessment within each of the data sets, similar with time to treatment discontinuation and time to next treatment being relatively consistent between a number of the different data sets that participated in this. When we looked at the outcomes associated with several of the demographic factors that were collected through this study, again, what we saw was the presence of some outliers, which may be due to the sizes of the population or those that were included in the population. We still saw relative consistency, at least in a couple of factors like gender or age when determined on a binary level, which I think points toward some potential uses for this. I think one of the things that we were very interested in this was, of course, there's a lot of discussion around what the appropriate regulatory potential for these type of data sets could be. And while that is certainly a valuable question to look at, one of the things we also wanted to look at was, for instances where this information isn't quite as exact, particularly in oncology where intermediate endpoints are frequently relied upon, could this type of assessment demonstrate longer-term clinical benefit where a randomized clinical trial, particularly in the pre-market, may not be feasible or even desirable for the sake of balancing access and time. What we saw here was of interest, I think, in looking at the binary breakdown between age in the population, ages 75 and older and 75 or below 75, was some relative consistency, at least with this type of drug, which I think is interesting since it always becomes a question of the patients that are included in the clinical trial and oftentimes may not be reflective of, say, the Medicare patient population, for example. So how much could this information be extrapolated toward other populations? And I know this is very difficult to read, but I think this was also what we tried to look at by even a further breakdown of some of the demographic characteristics. That while for these particular measures of age as broken down by decade, histology, and line of therapy, which may be a little bit more challenging to exactly extract from the electronic records. While these may not have exact correlation, I think it shows a very positive ability for this level of information to be extracted from these data sets and could be something that as the data sets grow over time could be very valuable in terms of looking at the different outcomes that are associated with different factors that otherwise wouldn't be included in the clinical trials. In this next slide, what we also wanted to do, in addition to looking at the potential for just extracting information from the different data sets, each of the partners performed the correlations between what they found in terms of the endpoints that were extracted based on real-world data, how that related to overall survival data. And while some of these correlations for instances like time to next treatment may not be overwhelmingly strong, they are consistent. And what we also saw for things like time to treatment discontinuation is that in many of these instances you saw a pretty high correlation between treatment discontinuation and overall survival. I should note that each of the partners, when they defined the parameters for drawing their data, we did include death as a discontinuation factor, so it's really not a huge surprise that there's a strong correlation in this instance, but perhaps here things like extracting time to treatment discontinuation from an electronic health record may be more readily accessible on a larger population than overall survival on a clinical trial population. And when you see relative agreement of the correlation, this may be a positive area that warrants future studies to determine the strength or the potential of this type of endpoint. And these bottom two are just representations of the information extracted specifically from those data partners that were utilizing electronic health records as their data source. One of the reasons we included this is I thought it was quite interesting that the assessment of progression-free survival as it correlates to overall survival was of pretty close similarity to treatment discontinuation, and PFS being a commonly used endpoint in many different oncology settings. Finally, just as sort of a non-statistical comparison, I guess, to put it, we really wanted to explore So how well did this information correlate in a quote-unquote manner, an unofficial type of calculation with what has been previously observed in clinical trials? So as I mentioned, for each of these data sets, it contains 3 different products that have been approved for this indication. We purposefully asked them not to separate this information based on drug. We didn't want to know, although I think that would be a very interesting question. I'm sure many other people would. But what this shows, sorry, let me try and go back one. What this shows here with the red dashed lines are the ranges that were observed between the pivotal trials for each of the agents under examination here. So a series of different phase 3 trials, and what we've put is the range of overall survival. And what you can see from each of the data sets is the extracted information from the real world data sets in most cases fell quite well sort of in line with this range of what was observed with the clinical trials, which I think was a very at least interesting finding because it's always sort of been speculated in terms of what the diminishing treatment effect might be observed given the way that oncology clinical trials are conducted in a very homogeneous selected population. There's been a range of estimates over the years in terms of whether that treatment effect it maintains itself once the drug is then put into a much more diversified population. And what we're seeing here, at least on this sort of side-by-side comparison of what was observed in the more restricted and refined clinical trials as compared to the more diverse real-world data set, is the outcomes in terms of overall survival have a relatively good overlay. Similarly here, in looking at time to treatment discontinuation, the dotted line in this particular figure depicts a study that was recently released through a collaboration with the FDA and Mass General Hospital, trying to look at these potential endpoints in the clinical trial setting. So they took a similar representation of the clinical trials for all of these immune checkpoint inhibitors in lung cancer and did an analysis on a patient level of their time to treatment discontinuation, the average of which is represented by the dashed line. And so what we saw again here with a couple of outliers, what we saw from the real world evidence, again in looking at both survival and as well as a more real world endpoint like time to treatment discontinuation, we saw relative similarity between what was able to be obtained from the real world data sets when comparing it to what was observed in terms of time to discontinuation from the clinical trials. So overall, what we found from this initial pilot sort of fact-finding mission was that there was a high level of shared characteristics between the different data sets, despite the varying sample size, the sources of the data, the size of the data sets, and they were able to be obtained in a very rapid fashion. We were able to show through this pilot that several extractable endpoints did correlate reasonably well with overall survival, which will require further validation to better understand what the utility of these types of endpoints could be, either for regulatory purposes, long-term value assessment, or whatever the different use cases may be. Interestingly, we found again that survival among patients that, as it was assessed through the utilization of EHRs, correlated relatively closely with what had previously been observed in the pivotal trials used to approve the drugs, as well as that there were a few characteristics in diving down to some of the deeper characteristics, such as age and sex, that correlated quite well between the different data sets that have been utilizing immune checkpoint inhibitors. So again, I want to thank the participants. This was a lot of work to pack into a very short amount of time. We definitely appreciate their collaboration and their expertise in driving this forward. We definitely look forward to identifying next steps, how we might take some of these endpoints, potentially validate them, look at ways that we might arrive at more standardized definitions in order to help this work move forward quickly, and look at different experimental techniques that might be able to be applied here to try and strengthen some of the evidence. So we really appreciate this work. It's been exciting to be a part of, and I look forward to discussing further as part of the panel.